Photosynthesis is a process by which green plants and some other organisms use sunlight to create food from carbon dioxide and water, shown by this equation here. Now, what does that actually mean? Plants have the ability to harness light energy, specifically from the sun, and convert the CO2 present in the atmosphere into empirical compounds of glucose, which can then be consumed by the plant itself or even other animals at different trophic levels. This is why photosynthesis is such an important process. Plants are able to be the autotrophs or producers and sustain the energy transfer of entire ecosystems. Let's break down this process. There are two main parts, the light reactions and the Calvin cycle. The light reactions, also known as photolysis, convert light energy from the sun into chemical energy that the cell can utilize. As the sunlight hits the chloroplast, the light waves enter photosystem 2, where the photons bounce around different chlorophyll molecules until they excite electrons that are accepted by the primary electron acceptor. However, those electrons are provided by the splitting of water and not carbon dioxide. The primary acceptor then passes the electrons down an electron transport chain located in the cytochrome complex, and much like the process of cellular respiration, the electrons are shuffled to molecules of higher electronegativities, each time allowing H plus ions to be pumped into the thylakoid space. This increases the proton concentration on one side of the membrane, which creates a chemical imbalance, more specifically known as the proton motive force. Then, by chemiosmosis, the H plus ions are moved back down through the ATP synthase enzyme, forming ATP from ADP and inorganic phosphate. ATP is the first form of stored energy in this process. But there's still more. The electrons that were moving down the electron transport chain still have to be accounted for. And after passing through the cytochrome complex, they enter photosystem 1, where a similar series of chlorophyll and electron excitation occur, until electrons are finally captured by another primary acceptor. This time, however, the electrons pass through a series of redox reactions, and the enzyme NADP plus reductase catalyzes the formation of NADPH. NADPH is the second form of stored chemical energy, and both the ATP and the NADPH are the chemical energy that will be utilized in the Calvin cycle.
The Calvin cycle builds carbohydrates from smaller molecules while consuming energy in an anabolic process from NADPH and ATP produced from the light reaction. It's important to note that glyceraldehyde 3-phosphate, or G3P, is produced from the Calvin cycle, not glucose. In order to create one G3P molecule, the Calvin cycle must occur 3 times, meaning that 3 molecules of carbon dioxide are required. A carbon dioxide molecule is first attached to a 5-carbon sugar called ribulose bisphosphate, or RUBP, and is catalyzed by the enzyme Rubisco. The molecule is unstable and is immediately split into 2 molecules of 3-phosphoglycerate. Each molecule then receives a phosphate group from ATP and electrons from NADPH to form G3P, a sugar. However, of the 6 G3P molecules, 5 must be regenerated to form RUBP, the 5-carbon sugar used at the beginning of the cycle. Through a sequence of reactions, 3 ATP molecules are used to rearrange the G3P into RUBP, now prepare to attach a carbon dioxide molecule and continue the cycle.
There are certain plants where the usual processes have been modified or altered to better accommodate the plant in response to hot, dry climates. 2 of these alternative methods are C4 and CAM plants, which have evolved to prevent dehydration. The goals of these are to minimize the wasteful process of photorespiration and to optimize the Calvin cycle. C4 plants have a specific anatomy along with their method of photorespiration, which includes mesophyll cells and bundle sheath cells. It maintains a CO2 concentration in the bundle sheath cells that favors photosynthesis over photorespiration. Mesophyll cells add CO2 to PEP to form a 4-carbon product oxalate before being transported to bundle sheath cells. From there, CO2 is released and enters the Calvin cycle. CAM plants, on the other hand, function by opening their stomata during the night and closing them during the day. During the day, they conserve water and prevent CO2 from entering, and during the night, they take the CO2 and incorporate into a variety of organic acids, which is stored in the mesophyll cells. Then, when light reactions occur during the day, the CO2 is taken from the organic acids to power the Calvin cycle.
Let's have a recap. First, photons from the sunlight enter the chloroplast, and the electrons provided by the splitting of water allow for the conversion of light energy into stored chemical energy in the form of ATP and NADPH. This stored chemical energy is then used in the Calvin cycle to convert CO2 into an empirical compound of glucose known as G3P. Remember, 3 molecules of CO2 are required to form one G3P.